Hello everyone, so I welcome you in this third series of lecture on renal physiology. So now we have saw that there is first process of glomerular filtration, then the next process will be reabsorption, and then there will be also tubular secretion. So all this will lead to urine formation. Okay guys, so now we are proceeding to the next step, that is reabsorption. We are done with glomerular filtration. So in reabsorption we will see each part, what all the things are that it absorbs. Okay, and then we will see tubular secretion. Although tubular secretion is not that much in detail, so we will see whatever part will secrete whatever substance consequently. Okay, so see, this is the thing which we need to focus now. We have done with glomerular filtration. Now we wish to see that what will be the contribution of PCT, what are all these ions, where are they reabsorbing. Then we will proceed to loop of Henle, then the DCT, and then collecting duct at the last. And then we will eventually finish entire urine formation, the core of renal physiology. Acid base balance has already been covered, and then what will be left is anatomy, that is micturition reflex. So let's proceed ahead. So basically now what we need to focus is on PCT. Okay, so PCT मतलब किसी भी पार्ट का सबसे पहला पार्ट है ना, मतलब अगर आप नेफ्रॉन की बात कर रहे हैं, सो पीसीटी इज द फर्स्ट पार्ट ऑफ द नेफ्रॉन. ओके, सो इट इज द मोस्ट इंपॉर्टेंट पार्ट. व्हाई? बिकॉज बेसिकली इट हैज हाई कैपेसिटी फॉर एक्टिव एंड पैसिव रीअब्सॉर्प्शन. ठीक है, तो इट विल बी very much important in reabsorption. Now what do you mean by active and passive? You know the basic terminology, but why? Because first of all, it has mitochondria. Okay, now if any cell has mitochondria, then it will be in a very good energy state, and it will have all the energy to do either active or passive. ठीक है. Second thing is that it have extensive brush border epithelial. Okay, now this apical side, I hope you are familiar with the terms. This is your PCT. So this here, your urinary side, this is your apical or luminal side, and then you have here basolateral side. Okay, so they have brush border epithelium on apical side, and they have many channels. Suppose sodium potassium ATPase channel, this will all together provide an extensive membrane surface area for absorption. Okay, so this is the thing, very basic. I don't need to give this thing to you right now, but if you read a book, you don't have to be demoralized that what is going on. Okay, so now we will see this process, that what are all the ions which are reabsorbing in this particular proximal tubule. You can see the figure 65%. So now in short, if I tell you, there will be three processes going on. Okay, ultimately you have to absorb sodium. So what you can do is you can absorb sodium via this sodium potassium ATPase channel. Okay, this is nothing new. I hope you know the sodium potassium ATPase channel normal physiology which you read in NEET times. Okay, first thing we will see this. Okay, second thing will be that sodium will be reabsorbed along with glucose. Okay, so this will be called as co-transport, है ना, sodium and glucose co-transport. Also there can be many other things which can be transported together like amino acids. Okay, so now this will be the second process. And what will be the third process is, this I have already covered in acid base physiology. You can see in this figure also. This is the third process, sodium hydrogen antipode. This is counter transport. This is co-transport, this is counter transport. Okay, so now if sodium will get absorbed, H plus ion will get excreted, and this is how we manifest acid base balance. Bicarbonate buffer, remember guys? So this is the thing, you have to absorb sodium, you have to remove H plus ion. So these three mechanisms will tell everything about your PCT. Now let's focus on this thing. So what is the reabsorption taking place at PCT? What happens? 65% of the filtered load of sodium and water and chloride. Okay, chloride का although 65% नहीं है, थोड़ा सा कम है, 60% के लगभग. But okay, so the main ions which you need to remember is sodium, water and chloride are reabsorbed by the PCT before it reaches the loop of Henle. Okay, so आधा काम यही कर देगा. This is cool. Now what happens is, first thing I have already told you, sodium potassium ATPase pump. It provides the major force for reabsorption of sodium, chloride and water throughout the PCT. Is that okay? Now what are the things? Basically, although sodium potassium ATPase pump is acting throughout the parts, but there is some difference. What happens is, there is difference in the absorption mechanism in early PCT and late PCT. Now let's see what are those differences. Okay, so now basically in the first half of your PCT, sodium is co-transported with glucose, है ना, glucose, amino acid and other solutes. I have shown you the image earlier. Okay, So sodium plus glucose first half. Okay. Now second half आते आते क्या होता है, basically in second half सोडियम ने ग्लूकोज इतना खींच लिया, सोडियम हैज अब्सॉर्ब्ड ग्लूकोज. सो नाउ इन सेकंड हाफ ग्लूकोज concentration will decrease in respect to chloride. Okay, now chloride will have an upper hand. So in the second half little glucose and few amino acids, but sodium is now absorbed with chloride ions. So is that okay for you? Okay, so let me bring that very clear. Second half of the PCT has chloride concentration 140 milliequivalent per liter. Early PCT have concentration of 105. Okay, so now what you have done, you have absorbed sodium with glucose in the first half. So now chloride has relatively become more. Okay, so this is the thing, and now you know that very well that now second part sodium will reabsorb with chloride. Is that okay? So this is all about reabsorption in PCT. Now we will see secretion by the PCT. So I hope things are clear to you. Say with me, what are the things which you need to know at the reabsorption of PCT? Just reabsorb 65% of sodium, glucose, water, everything, है ना. क्लोराइड के साथ भी कराएंगे. फर्स्ट हाफ में सिर्फ सोडियम ग्लूकोज को ट्रांसपोर्ट, सेकेंड हाफ में sodium chloride co-transport. Okay, and the sodium hydrogen antiport, that the counter transport is a little fraction and that is operating at its level to maintain the bicarbonate buffer. Is that okay? Now let's proceed to secretion. So what will proximal tubule secrete? It will secrete organic acids, bases such as bile salt, oxalate, urate, catecholamines. All the things which are excess in the body or needs to be expelled out are excreted at this level. Okay, what is important for you to remember is penicillin. Okay, now this is very important because in pharmacology whenever you will study there are certain long acting penicillin and you have to search right now on Google that what do we combine with penicillin. There is a particular substance due to which it also starts with P, due to which you can decrease the clearance. Okay, because penicillin is rapidly cleared by the kidney because of this proximal tubule. जैसे ही penicillin आएगा proximal tubule बाहर फेंक देगा, तो आपका पेशेंट पे असर कैसे होगा. ओके, इफ यू वांट टू गिव पेनिसिलिन एज एन एंटीबायोटिक टू द पेशेंट, यू नीड टू हैव इट इन योर बॉडी सो दैट इट कैन प्रोड्यूस एन इफेक्ट. But your PCT is doing an evil role by excreting it out rapidly. So you need to combine with other substance. So this is the correlation of physiology, okay, with pharmacology. Now the third thing is PAH. Okay, para amino hippuric acid. This is again rapidly secreted by PCT. And it is so rapid that approximately 90% of the PAH is secreted out, excreted in urine. Okay, so if I will tell you, we will see further if possible that what is clearance and how to measure GFR and all those things. So I hope you remember that PAH is an important term there in GFR calculation and the principle is again the same. क्योंकि अगर आपने जितना दिया उसका 90% आ रहा है तो लगभग वैल्यू GFR के बराबर होगी, रीनल प्लाज्मा फ्लो के बराबर होगी. So this is the thing which you need to know about proximal convoluted tubule. I wish that it will be sufficient at all the levels. Just remember those high yielding diagrams. Okay, I will revisit them to you. Okay, first of all bicarbonate buffer, then your co-transport and counter transport, and then what you have is the main junk, main gist, that is sodium potassium ATPase pump. So I hope things are clear with you. Okay, so now let's proceed to the question. What is the question that you need to answer in the comments? Okay, so you need to take some efforts and you have to go on the Google and search what is the long acting penicillin. Write down in comments, and if you wish you can contribute. Thank you so much guys. Stay safe, stay healthy. We have completed 130 plus videos. Kindly go to the playlist section and start watching as much as you can. Thank you so much. Stay safe, stay healthy and keep learning.